Hi everybody, welcome back. It is your AP Biology teacher, Mr. Poser. Here it is. It is the Unit 6 recap. This is perhaps the most requested video that I've ever had before, which is fun. But we got a lot to cover in this video, and I think this is probably, in my opinion, I don't like to say like, oh, this unit is harder than the others, but there's a lot to this one, and there's a lot of technical terms that you can get bogged down by, that my own students have gotten bogged down by in the past. And I think it's really important to know the general concepts and the general ideas behind gene expression and all that before in a replication, for example. You can get really bogged down into details. So I think it's really important to know the general information and the overall processes of how these work before you get into this nitty-gritty specifics of it. And that's what I try to emphasize here in this video. So in more detail, okay, I cover all these topics in my Unit 6 playlist. So if you haven't checked those out, check those out. But here it is. I'm going to try and wrap up this entire big unit in one video. I'm going to try to stay down to the script as much as I can and not go off topic too much. But here we go. Here are the topics. 6.1 is on DNA and RNA structure. 6.2 is on replication. 6.3 is on transcription and RNA processing. 6.4 is on translation. 6.5 is on regulation of gene expression. 6.6 is on gene expression and cell specialization. 6.7 is on mutations, and 6.8 is on biotechnology. Here we go. Unit 6. This is a doozy here. 6.1 is kind of a review of 1.6, in my opinion. It's about DNA and RNA structure, the differences between them in terms of their nucleotides, in terms of their overall shape, and in terms of their function, kind of. But anyway, this is a continuation of genetics in a way. This is a field of biology that's called molecular biology, and I defined it down there. It explores how cells use genes as instructions to carry out functions and produce traits. Okay, where do you get your genes from? Well, they are passed down from your parents, and that's what our last unit was about. Unit 5 was about heredity, and this one is getting into how those genes are actually used within a cell to produce traits. So we're getting into the specifics of how DNA actually works. Unit 5 is all about where you get it from, and then the patterns that go along with that and the probabilities that go along with that. This is how it's actually used. And this unit is about gene expression, the process by which segments of DNA, aka genes, are decoded and used to make particular proteins within a cell. How a cell uses DNA to make proteins, like I was just saying. Expression has to be regulated, hence the title of this unit, gene expression and regulation. Cells needed to determine when and where to produce proteins based on their overall function. So this is all about making proteins. DNA is the instructions that cells use to make proteins. That's true for all living things. All right, but you can't be expressing all of your genes all the time. You can't be making every single protein. So gene expression has to be regulated, and that's something that we explore, especially in topic 6.5 in this unit. But here it is, the overall process. Here is DNA. Transcription is making mRNA, messenger RNA, using a small piece of that DNA called a gene. Then it makes what's called a transcript of mRNA. And then that mRNA is decoded and translated by a ribosome and special molecules called tRNA, another type of RNA, to build an amino acid sequence that will eventually be folded and become a protein and produce traits, okay, and result in cell specialization. So here it is, very, very simple again. DNA and RNA are the genetic material, and it's all life speaks the same language of A's, T's, C's, and G's. In DNA, which is amazing. DNA is inherited from parents. It's a segment of DNA or a discrete unit of DNA is called a gene, and this is what gene expression is all about. So keep that in the back of your mind, that we're all about going from DNA to RNA to proteins, all right? That's the whole thing of this unit here, okay? Don't get too, don't lose sight of what this unit is about, because it's easy to do that. I can't stress that enough. So DNA and RNA contain the instructions to enable organisms to produce proteins from one generation to the next. So really what you get passed down, or what you pass down to your kids and what your parents pass down to you are instructions. It's a blueprint of living things. This is how you build a living thing, hence genetics. That prefix means the creation of, hence like genesis or generation, right? This is the instructions to build life. An organism's DNA comes from chromosomes inherited from previous generations. And again, that's what our last unit was all about. And here's another picture of gene expression. DNA in the genome is transcribed through to RNA, and RNA is translated into a protein, right? And what do you actually inherit from your parents? You would actually inherit through chromosomes. We talked about that last unit as well. Are made of DNA and a protein complex called chromatin. Prokaryotes have circular chromosomes. Eukaryotes have linear chromosomes, and this is what actually gets passed down. Chromosomes are made of basically proteins called histones that DNA is wound around, kind of like a spool of thread a little bit. Lots of different spools of thread that are kind of stacked on top of each other, and DNA is wound around each of them. Because one piece of DNA could be really, really, really long, but it has to be packed tightly into a nucleus in a eukaryote. But in a prokaryote, okay, the chromosomes are circular, okay, and prokaryotes also have little pieces of DNA called plasmids, which aren't really chromosomes, but they're much smaller pieces that are in a circle, and they're transferable from one cell to the next. And that's going to become important later on, so hold that thought. Remember plasmids. Okay, DNA and RNA are both what we call polynucleotides. Poly meaning many, nucleotides meaning, well, those are the monomers of DNA. They are chains of nucleotides that consist of 3 portions, and we talked about this in 1.62. A nitrogenous base, that could be adenine, guanine, cytosine, or thymine, a 5-carbon sugar, ribose in RNA, and deoxyribose in DNA, and a phosphate group. Okay, so here's your 3 parts right here. Phosphate group, bang. 5-carbon sugar, ribose or deoxyribose, bang, right there. And then here's our base, all right? And now DNA is just thousands and thousands and thousands of these molecules put into 2 different chains and twisted around. And that's the structure of DNA. Now, in other AP Bio classes, you may, depending on who your teacher is, you may or may not have spent a lot of time studying how we discovered that DNA was the genetic material, or how we discovered the base pairing rules, or how we discovered this overall structure of DNA, and that it was a double helix. So you might be talking about experiments like the pulse chase primer DNA replication, where we found out that DNA replication is semi-conservative, or the studies of Watson and Crick and Rosalind Franklin and Chargaff and that kind of stuff. We're just kind of going to gloss over that because it doesn't explicitly mention those in the AP Biology course and exam description, so I kind of just gloss over it like, okay, this is how we kind of discovered it a little bit, now moving on. I'm just going to skip that entirely in this video. So if you're wondering for where information is about like the pulse chase primer experiments, or I can't even remember one of the other ones, okay, I'm not going to cover it here, so just a little disclaimer there. But anyway, nucleotides can be categorized into 2 groups. They can be pyrimidines or they can be purines. Pyrimidines are nucleotides with 1 ring of a nitrogenous base. Okay, so here's a pyrimidine. Cytosine is one, thymine is another one, and uracil in RNA is a pyrimidine. And purines are nucleotides with 2-ring nitrogenous bases, so like adenine and guanine here. You can see 2 rings in their nucleotide base. All right, so those are pyrimidines and purines. Remember the difference between those 2. DNA's overall structure consists of 2 polynucleotide strands that form a double helix or a twisted ladder shape. So if you take a ladder and imagine just going like this with it at either end, then you arrive pretty close to the overall shape of a DNA molecule. RNA is very similar, except it's only 1 strand, as you can see in this picture here. But in a DNA molecule, hydrogen bonds bind cytosine to guanine, and they bind adenine to thymine. One of the things that all of my students remember in every single year of AP Biology is that the apple goes in the tree and the car goes in the garage. So if you remember that, you can remember your what we call your base pairing rules, your Chargaff's rules, that A always pairs with T and C always pairs with G when it comes to the 2 polynucleotide strands of DNA. Apple goes in the tree, car goes in the garage. Everybody remembers that. So here we go. RNA, again, difference between RNA and DNA is that RNA has 1 polynucleotide strand while DNA has 2, and RNA contains uracil as a base while DNA contains thymine. Okay, so in this case, if we're matching up nucleotides, apple goes underwater. I don't know, starts with a U, in the umbrella, I don't know. And C still goes with G. All right, but T on a DNA molecule will match with A, and we're going to talk about that in a second. Last difference between DNA and RNA is that DNA nucleotides contain deoxyribose, while RNA contains ribose sugars. Deoxyribose lacks in what's called a hydroxyl, or an OH group, on the 2' carbon. So here's a very clear difference between deoxyribose. So why do we call it deoxy? Well, because there's 1 less oxygen here on the 2' carbon. Here's the 5' carbon, here's the 3' carbon, here's the 2' carbon, and that's another difference between DNA and RNA. And again, we covered that in 1.6. Okay, last little piece about DNA structure is that DNA strands are what we call antiparallel to each other. This is a kind of geometry, mathematical term. 2 strands are oriented in opposite directions, and they're upside down to each other. Okay, so here's one DNA strand. If it's facing this way, the other one is parallel to it, but it's facing the other way. Hence, they are anti-parallel. Now, we use directional terms for DNA based on the number of the carbons in the deoxyribose sugar. We call one the 5' end, and we call one end of the DNA molecule the 3' end, because that's kind of how these deoxyribose molecules are oriented, right? So this is a 5-carbon sugar, right? You can count 1, 2, 3, 4, 5. So if we count from this way going down 5, 4, 3, 2, 1, you can figure out that this is the 5', this is the 4', this is the 3'. All right, so that means that this deoxyribose, if it was part of a nucleotide, would kind of be facing this way, while the other one kind of faces the other way or runs into 3' to 5' direction. And you can kind of see that here, right? So if let me take my marker here, for example. Okay, I would know this is the 3' end of the DNA, and this would be the 5' end over here. Boop. Right there, and this would be the 5' end of the DNA molecule, and this would be the 3' end, and I can tell by those carbons and where they are oriented in those deoxyribose molecules. Okay, but that'll be it for 6.1. If you want to check out more details about 5' to 3', again, I got videos on those, but I'm going to move on because this is a long video. 6.2, DNA replication, super duper important. It is the copying of DNA. Life cannot exist without DNA copying itself. We can't live forever, so we have to copy our DNA and pass it down, right? And so DNA replication is an extremely important process for all living things, and that it guarantees the continuity of life, right? Because we don't live forever, we got to pass down our DNA. We got to pass down our instructions to our offspring. So this is a very important concept, okay? The base pairing rules, A to T, C to G, apple goes in the tree, car goes in the garage, enable replication. And again, there's a whole series of, studies that particularly pulse chase primer experiment that you may or may not have studied in AP biology. That's going to explain how scientists discovered that DNA is semi-conservative through replication. But we're going to skip over that. But what you got to know is that DNA replication is semi-conservative. 2 daughter newly synthesized molecules are made of 1 parental and 1 template strand, right? So how DNA replication works is that these 2 old strands, right? If we're trying to make a new copy of this molecule, okay, we take the old strands apart from each other and we build this new strand based off of the old ones. So we don't have a brand new DNA molecule, right? We have 2 brand new strands that are kind of attached to the old strands right here. So hence semi meaning half conservative. Half the DNA molecule is saved from the previous copying. Okay, it can be summed up in 3 steps. DNA is unzipped. Okay, it's just take apart those 2 DNA strands and it's stabilized. DNA polymerases, 2 enzymes that are going to be producing new DNA nucleotides, synthesize new strands based on the old strands, and they work in 2 directions. Hence DNA's anti-parallelism. And ligase joins the fragmented DNA strands, what are called Okazaki fragments, into 1 continuous strand. Now, I need you to get those 3 steps. Okay, unzipped, DNA is copied, new nucleotides are formed, and then the little pieces called Okazaki fragments are sealed together. 1, 2, 3 basic steps of DNA replication. Because there's a lot of terms here, and there's a lot of enzymes that you can get bogged down by. And my students tend to get pretty confused by this. So stay on it. Okay. Initiation of replication. This is step 1. Helicase is the name of the enzyme that unzips the double helix and opens up the replication fork. So here's helicase. It kind of just wedges in there and splits apart the 2 DNA strands. It breaks those hydrogen bonds between those nucleotides. And it forms what's called a replication fork. And you can see a fork in the road right there, separating parental strands of the DNA. Two proteins that are going to be stabilizing and making sure that this DNA strand stays open and it's not completely messed up. Single-strand binding proteins prevent strands from rejoining and just forming back together. And a special enzyme called topoisomerase stabilizes the DNA and prevents what's called supercoiling, right? If you split apart these 2 strands, it's going to get extra twisty upstream. Yeah, and you can use that on your test. Say extra twisty upstream of the where the replication fork is, and there's an enzyme called topoisomerase that prevents that from happening. Step 2 here, before DNA polymerase synthesizes new nucleotides, okay, RNA primers need to be placed on the template strands. Now, DNA polymerase needs a place to start. It's going to add new nucleotides based on the old strand. It's going to do its job, but it needs a place to start, and that's what primers are for. Prime means start or first, right? So that's what you can remember. Primers have to go first before DNA polymerase can go. Primers serve as a starting place for DNA polymerase, and they're added by what's called a primase enzyme. It's very appropriately named. So primase adds primers and allows a place for DNA replication to begin, or DNA polymerase to place new nucleotides. After that, DNA polymerase adds new nucleotides only in the 5' to 3' direction. That's something to remember once it binds to the primer. Okay, so here's a primer. DNA polymerase is going to attach itself near the primer or onto the primer so that it can start producing new DNA nucleotides. And primers are made of RNA, so that RNA eventually needs to be replaced. But DNA's antiparallel structure and the fact that DNA polymerase only works 5' to 3' creates complications, and this is where DNA replication gets a little complicated. Okay, because it only works 5' to 3', and DNA is anti-parallel. That means that one strand can move forward or be copied in the forward direction, but the other strand has to be copied in the backwards direction. And the strand that's moving in the forward direction or toward the replication fork, it has an easy job. That's called the leading strand, right? So I don't have this as a moving picture here, but helicase is going to keep moving down through that DNA molecule. It's going to keep spreading those the template strands apart, okay? And DNA polymerase here on the leading strand just has to follow the leader, all right? And it can continuously make this new strand of DNA. But here's the other thing. You only need one primer for the leading strand, but, right, since, DNA is anti-parallel, this one's moving forward, but this one has to move backwards, and hence that's what's called the lagging strand. And these two strands are synthesized at the same time. And I said, leading strand is in the replication of the replication fork. It just kind of follows helicase down, but the other one, okay, has to go in the opposite direction because 5' to 3' is the only direction that DNA polymerase could work in. It has to work backwards, so multiple primers are needed, right? So helicase is moving this way, okay, if you can see my cursor. Helicase is moving this way. DNA polymerase on this strand is moving that way, so that means that the more DNA, the more DNA that helicase, opens up, right, the more the DNA polymerase has to start over again and again and again. That means we need multiple primers on the lagging strand, and we need to copy it piece by piece by piece by piece. Again, I explained this a little bit more in my 6.2 video if you want to check that out. There's some videos and some GIFs that will show you oh, here's how this works backwards and this works forward. But the lagging strand is thus only built one piece at a time. Each of these pieces are called Okazaki fragments, and here are the Okazaki fragments. Here's kind of what I was talking about again. It's not moving, and I think it's best to be explained while it shows that it's moving. All right, but helicase is moving this way. Leading strand can just follow along with helicase, but the lagging strand has to work backwards, right? So as helicase moves down here, and there's topoisomerase, as helicase moves down here, all right, it's got to start over a bunch of different times and place a new primer, work backwards, place a new primer, work backwards, place a new primer, work backwards. Hence, it does it piece by piece, and those are what are called Okazaki fragments that are formed. And a special enzyme called ligase is going to seal those Okazaki fragments together to form one continuous piece of DNA, one continuous polynucleotide strand. And there you have it. And then once that's done, DNA, you got 2 copies of it, and you can pass down your genetic information and continue life on Earth. Fabulous. All right, so here's how DNA is used, right? This is the overall picture again of the whole unit. This is called the central dogma of molecular biology, and this is as simple as you could put it. All living things have information stored in DNA as genes, which provide cellular instructions for making proteins. All right, so genes are the instructions to make proteins, but there's an intermediate step, and it's called transcription. I'm going to get to that in a second here, but the process by which, I'm going to say this a bunch of different times, gene expression is the process by which genes are used to make proteins. But DNA does not directly make proteins, right? So there needs to be some kind of intermediate step, and that's called transcription. And that intermediate is RNA. Transcription provides this intermediate step in the process of gene expression. RNA is synthesized using code from DNA. That's what transcription is, okay? RNA polymerase, very similar to DNA polymerase, but not the same, right, because it's RNA, uses a single strand of DNA to synthesize new bases in an RNA molecule. And there are 3 types of RNA. messenger RNA, which is probably the one that we're going to talk about the most, transfer RNA, and ribosomal RNA. And we're going to talk about each of those over the course of this unit. And there's actually one more, but we're going to get to that later on. But RNA polymerase is going to make RNA. DNA polymerase makes DNA. RNA polymerase is going to do transcription. DNA polymerase is going to help with replication. And it's not the same, though. Transcription makes RNA from a DNA strand. Here's RNA again. Our replication makes DNA from DNA strands. Transcription is used in gene expression, right? The actual using of the genes to make a protein. That's what RNA is used for. Transcription is used for. Replication is used before cell division to, pass on its DNA, pass on its instructions. And that replication is used for passing on the instructions. Transcription is actually for using the instructions. Both transcription and replication involve the base pairing rules, except if you remember from 6.1, RNA has uracil while DNA has thymine, right? So those U's replace those T's. And once again, there are 3 easy steps to transcription: initiation, elongation, and termination. Those are your 3 easy steps of transcription. Step 1, initiation, involves the RNA polymerase binding to what's called a promoter. A promoter is a section of DNA that's going to say, hey, this is where you start transcribing right here. RNA polymerase, you bind right here. And then promoters, I already put that right there, okay. Promoters are regions of DNA where RNA polymerase binds to begin transcription, and transcription factors control the binding of RNA polymerase. Can be negative, where they can prevent transcription, or positive. So they're extra proteins that will tell RNA polymerase, hey, you should bind here, or hey, don't bind here right now. We don't want to express this gene. So transcription factors, they take all sorts of different shapes and sizes, and it gets pretty complicated, but we won't get too far into that just yet. So yeah, that's the first step. RNA polymerase binds to the promoter along with its transcription factors. Elongation, step 2, RNA polymerase slides along the DNA template, kind of like DNA polymerase, okay? Kind of like DNA polymerase, slides along the DNA template strand, pairing DNA and RNA nucleotides. For every A, it adds a U. For every T, it adds an A. For every C, it adds a G. And every G, it adds a C. Same as before, except with the U, right? So it creates this mRNA strand, one strand that we call the primary transcript. And how does it end? Through termination. A DNA sequence called a terminator causes RNA polymerase to detach and release the mRNA transcript. All right, so here's here's RNA polymerase. It's going to create this mRNA molecule based off the old template strand. When RNA polymerase finishes its job, it creates what's called the primary mRNA transcript. Okay, primary means first. It's not done, right? There's got to be some things that happen to it before that RNA gets shipped out of the nucleus and sent to the ribosome to start to make a protein, right? It has to be modified and processed before it's translated. And there's 3 things that has to happen. It has to have a 5' cap, a 3' poly A tail, and it needs to be spliced. Okay, so 3, this is what we call RNA processing. This is a big step in gene expression. 5' end of a primary transcript receives a modified guanine, and this kind of stabilizes it and makes sure that the primary transcript doesn't get degraded by the parts of the cell before it can be expressed. But sometimes we'll get to it. Part 3' end receives a long chain of adenine nucleotides, hence the poly A tail, which helps with mRNA export and ribosome attachment, right? So the poly A tail allows it to be shipped out of the nucleus and sent to the ribosome, and it can attach to the ribosome so it can be translated. And that's what the poly A tail does. And those 2 things have to happen to your mRNA before they get translated. And the third thing that has to happen is what's called splicing, removing non-coding segments, which are called introns, and connecting the coding segments, exons, of the primary transcript. Now, this is something to remember. Introns are, okay, I actually taught this wrong one time. I said introns are kept in. No, that's not true. Introns are spliced out, and exons are kept in of the primary transcript. So check it out. Here's a primary mRNA transcript. We have exons and introns here. Check it out. The introns, after a spliceosome comes through, it cuts out or edits a transcript. It takes out the introns and leaves in the exons. So here's a primary RNA transcript, and here's what we would call a mature mRNA molecule. There's a 5' cap, there's a poly A tail, and the introns have been spliced out. This is RNA that's ready to be translated. Alternative RNA splicing, which we're going to talk about later, can lead to the production of different polypeptides or amino acid chains from a single gene based on what is treated as an intron and what's treated as an exon. You can get different things from it. Here we go, 6.4, right? Here's ours, mature mRNA transcript. It is now ready to be sent to the ribosome and start to be translated. And here we are. 6.4 is all about translation. Sequences of DNA nucleotides are like letters in a language. Those A's, T's, C's, and G's are like letters in a language. So imagine speaking a language that's only made up of 4 different letters. Imagine trying to make a video like this or write a paper or something using only 4 different letters. All right, there's got to be a lot of different letters in order to send your message and make your point. So DNA sequences are very, very long, right? DNA messages are transcribed into mRNA, then the words in the code are translated, and that's what this is about, into 20 different amino acids. Yes, there are 20 different amino acids and 64 different what are called codons, groups of 3 bases, right? Each of these words are 3 letters long, code for 20 different amino acids. So here's gene expression again. DNA, transcription into RNA, translation into proteins, and we are at this second step right now. Okay? And what is this wheel of fortune over here? This is what we call a codon chart, and I'm going to show you what that is in just a second. The coding region of the mRNA begins with AUG, which is called the start codon, and which codes for what's called methionine, an amino acid, and that's what will begin our amino acid chain. So let me go back for a second. Here's our mature mRNA molecule, right? The first translated region is going to start with an AUG sequence, and that's what we call a start codon. And that will tell the ribosome to make a methionine amino acid. And then all the groups of 3 following that AUG are going to tell the ribosome to assemble a very specific amino acids together in order to make the right protein with the right amino acids. Because if the amino acids are not right, then the protein's not going to work the same way. And we're going to talk about that later on too. But codon charts are easy ways for us to convert codons into specific amino acids. My mouse pad over here is a codon chart. I'm very proud of it. But here's a codon chart. 3 codons are what are called stop codons, which stop a ribosome from adding more amino acids to a growing polypeptide chain. So you can convert these groups of 3 mRNA nucleotides into words, into amino acids, and you add the right amino acids in place. So here's a codon chart again. All right, how did I find that AUG codes for methionine? Well, the first letter in AUG is A, right? And you can see this just doesn't say guac, right? Even though guac is delicious. The first letter is A, right? So I have this first circle. I can find A. You can kind of put your finger on it. That's what I tell my students to do. Put your finger on A in the middle. All right, the second letter is U, so you go to the second circle and follow A to U. And then the third letter is G, so you follow that to the third circle. You go A to U to G, and then here's methionine. And you can see that it's also called a start codon, right? And here's some stop codons. So UAG makes a stop codon. UGA makes a stop codon. UAA is also a stop codon. But all these other ones, right, are coding for different amino acids. And that's how you kind of use it. And again, if you need more practice with that, watch 6.4, go to my video. Translation of mRNA codons into amino acids are carried out by ribosomes and what's called transfer RNA, tRNA. Here's our second type of RNA. We talked about mRNA, here's tRNA. tRNA carries specific amino acids to the ribosome that matches with the mRNA codons using anticodons, right? So think about this, right? If I have AUG as my mRNA codons, okay, what tRNA is going to do, it's going to have what's called an anticodon that is complementary. It matches up with AUG, and it's going to carry the specific amino acid methionine, that met, okay, and it's going to match up with it, right? So it's going to have the anticodon for AUG is going to be UAC. And notice I'm still using U because RNA has U, right? And there's my crappy handwriting there. But yeah, that's what an anticodon is, right? It's going to have, every tRNA molecule is going to have an anticodon that matches up with that mRNA codon and a specific amino acid to carry. Ribosomes are made of 2 subunits of rRNA. Here's our third type, ribosomal RNA, through which mRNA is fed kind of like a sandwich, right? So here's your, it's silly, right? But here's a ribosome. It's got 2 subunits, and kind of mRNA is kind of slid through here. It's kind of like fed through like a kind of like a copy machine in a way. Or if you have ever checked out the old Scantron machines, that's kind of what I think of. But Scantrons are like, those are ancient now, right? But anyway, and here's the amino acids. Here's the tRNAs that are carrying the amino acids. And you can see they add 1 amino acid at a time to a chain. And this is how translation works. tRNA anticodons complement mRNA codons and are written in the opposite direction. So as I showed you before, AUG goes with like UAC, right? Each tRNA carries a specific amino acid that grows to adds to a growing chain, which is going to become a protein. And a stop codon triggers a release factor to release the polypeptide from the ribosome. So check it out. Here it is once again. Here's our ribosome, our RNA, 2 subunits that messenger RNA gets fed through. And here come tRNAs. They bind to and they complement an mRNA sequence and bring in a specific amino acid to grow to a peptide chain in order to build a properly functioning protein. Now, kind of left turn here, we're going to talk about viruses. What? Viruses? Yeah, like because viruses follow all the same rules of gene expression. And they basically hijack how a cell does gene expression to make copies of itself. Why? It's not a living thing. It's an infectious particle that consists of only genes and a protein coat that uses a cell's molecular machinery to express the viral genes. It doesn't qualify as a living thing. It can't metabolize. It can't make its own DNA. It can't make its own protein, so it tells a cell to do it. It takes over a cell's ability to do that. And we're not going to spend too long on this, but like I said, viruses are just a protein capsid with antigens for finding and latching to a host cell and some DNA or RNA. It's basically following the rules of gene expression, except, hey, cell, you're not going to make your own proteins, you're going to make my proteins now. That's what that virus is. I am the captain now. And it tells the cell to copy that DNA and express its genes to make more viral proteins and produce more viral DNA so that it can make more viruses. Yay! Sickness! And viruses attach to a host cell and release genetic material, which is then copied, transcribed, and translated to make more viruses. So everything that we've talked about so far, replication, transcription, translation, a virus does those, or I shouldn't say that, a virus makes a cell do those to make more viruses. I think it's absolutely fascinating. Viruses are really sad and like scary, but they're also really, really cool. Especially retroviruses. These are especially very sad, but also very interesting. They are viruses with the ability to convert RNA into DNA that is then integrated into the host cell's genome. What? It's going backwards. Yes, reverse transcriptase is the name of the enzyme that allows a retrovirus to turn RNA into DNA and then take that DNA and put it into the host cell's genes, into its genome, into its nucleus. So that host cell might still be healthy and this viral DNA might lay dormant for a little while. And that cell is going to copy all of that DNA and make copies of itself, right? And all those other genes are still going to have that viral DNA. And it's going to have the ability to make more viruses. So human immunodeficiency virus, or better known as HIV, is a well-known retrovirus that breaks the rules of the central dogma. Somebody who has human immunodeficiency virus, HIV, somebody who is HIV positive is they are HIV positive for life. And this is why, because the initial virus, right, that infected that person integrated its genome, its genes into that person's cells, into that person's genome, into that person's DNA. So that sticks with them for the rest of their lives. Now, this is not by any means. This is a big deal. Like 40 years ago, in the 1980s, human HIV and AIDS, and it still is a big deal. Don't get me wrong. Do not get me wrong. It is still a big deal. But we've made really great advances in medical tech that allows people who are HIV positive to live long, healthy, happy lives, which is an amazing thing. So this is a virus that can be lived with, but that's why it's there for somebody's life, because of reverse transcriptase and it being a retrovirus. All right, a couple more topics to go. Here we go. 6.5 is about the regulation of gene expression, controlling how and when and where and why genes are expressed. Gene expression involves the transcription of DNA into mRNA, then the translation of mRNA into amino acid chains, which are folded into protein. We know that. We've been over it. All living things have to control what proteins they synthesize. Not every gene can be expressed at full blast all the time. That doesn't make any sense, and that's not going to help anybody out, especially a multicellular organism like ourselves, right? It's not going to help anybody. You might not be in the right environment to express certain proteins. It doesn't make sense to do that. Like if you're a bacterial cell and there's no lactose available to break down and digest in your environment, then you're not going to express the genes that break down lactose, right? We're going to talk about that in a second. If you're a nerve cell, if you're a brain cell, one of the 86 billion ones up here, you're not going to be producing digestive enzymes. That just doesn't make sense, right? So gene expression needs to change in response to environmental changes. This is true for both eukaryotes and prokaryotes. All of them things have to be able to do this. Now, prokaryotes, this is how they do it. They do through something called operons, which are segments of DNA with different components that coordinate the production of proteins. And this is what an operon looks like. I don't know why promoter isn't spelled with an E there. But operons, what they do is they regulate production of enzymes in response to environmental changes. And that example of lactase that I just talked about before, this is the most well-studied way that prokaryotes control their gene expression. Okay, and this is what's called the lac operon, lac short for lactase, right? Regulates the production of lactase, which is the enzyme that breaks down lactose, when glucose is unavailable in the environment. Here's the lac operon. It is a group of genes that's going to work together to control how much an enzyme like lactase or beta-galactosidase is produced. Ready? This is how it works. There are several different segments here. Number one, we've already kind of talked about the promoter. So LAC-P here is what we call the promoter, and that's the DNA segment where RNA polymerase binds. Now, the other parts of this operon are going to control how and when RNA polymerase will bind to the promoter, okay, or whether or not it can slide down from the promoter and actually transcribe the rest of the genes, right? But we already know what the promoter is. We talked about it. It's where RNA polymerase binds to transcribe that gene. The operator is the segment right next to the promoter that acts as an on or off switch, and it controls DNA. When the protein called the repressor, I'm going to circle that down here as well, protein called the repressor is bound to the operator, guess what? It represses or stops that gene from being transcribed. It stops RNA polymerase from sliding down here to LAC-Z and LAC-Y, and I can't see what it is, and LAC-A in order to make these enzymes because the repressor is in the way. Hence, the gene is off. Now, the gene is on when the repressor is out of the way. And how do you get the repressor out of the way? Well, I'm going to talk about that in a second here. The LAC genes, LAC-Z, LAC-Y, and LAC-A, are coding regions where RNA polymerase transcribes and produces lactase enzymes. So RNA polymerase, when this gene is turned on, it's going to go whee, slide down the genes there, okay, and it's going to start to transcribe. There's our mRNA, and it'll be translated eventually into those enzymes, right? LAC-Z, LAC-Y, and LAC-A, very important for lactase. Now, there's one over here that we haven't talked about yet, LAC-I, which is what's called a regulatory gene. Regulatory meaning controlling, right? And it codes for what's called the LAC repressor, what I talked about before, right? So when this gene is expressed, it produces the LAC repressor, and it's going to turn off. The repressor is going to bind to the operator. It's going to go whoop. It's going to bind to the operator, and it's going to turn that gene off, right? But when the repressor is not there, okay, then that gene is on and can start producing lactase. And how do you turn the gene on? Well, you do that through what's called an inducer, right? And an inducer in this case is lactose. So when lactose is present, this is why this makes so much sense, because when lactose is present, you want to break down lactose to make glucose with it and make ATP with it, right? You want to break down that sugar that's in your environment. So this is what makes so much sense about it. When lactose is present, it turns off the repressor. Okay? It inactivates the repressor. So what it's going to do is going to turn on the gene, right? If the repressor is there, the gene is off. Or lactose, what it does, it binds to the repressor and removes it from the operator, the on-off switch. And therefore, this gene is turned on, and it's going to start making lactase to break down the lactose that's in that bacterial cell's environment. So this is what's called an inducible operon. And here it is again, allolactose, which is a lactose isomer, or just lactose itself, inactivates the LAC repressor and turns the gene on, allowing RNA polymerase to transcribe the lacteins and make lactase to break down more lactose, right? And the name of this is what it's called, an inducer. Lactose removes the repressor from the operator. And again, if you need to, check out what those terms are, go back a little bit. But lactose turns off the repressor, gets the repressor off of the operator so that we can make more lactase. And that's what's called an inducible operon. Another one, you don't have to know specifics about it. Other operons like the trip operon or what are called repressible, which is kind of the opposite a little bit. When tryptophan, which is an amino acid by the way, is present, that means the gene is turned off, right? So lactose is inducible because when lactose is there, the gene was turned on. But tryptophan, when it's present, turns the gene off. So that's what's called repressible. As I said, the trip operon is turned off when tryptophan is present. Okay? Now, eukaryotes also have to control their gene expression. It's super important that they do because it results in differentiation of cells, right? So bacteria are not really differentiated. They're just free-living organisms. They have to control their gene expression based on what's in their environment. Eukaryotic cells, okay, like the ones in us, right, multicellular organisms, they have different jobs to do, okay, and they have to control their gene expression because it doesn't make sense to make proteins that won't allow them to do their job. Like I said before, it doesn't make sense for a neuron to produce digestive enzymes. It doesn't make sense for a muscle cell to produce, I don't know, keratin, okay, in your skin or something like that. So eukaryotic cells are differentiated. They have specific functions in multicellular organisms. All your cells have different jobs to do, and they express different genes in order to do that job. Cells have the same DNA, but receive signals to express different genes, leading to specialization. So like your stomach lining cells, the DNA in their nucleus is the exact same as the DNA in the neurons in your brain, right? But they're not doing the same thing. They have the same genome, but they're doing different jobs because, and this is where this comes from, because they're expressing different genes. So here's all the different ways that eukaryotes control gene expression. And I like this picture. I blew it up because it shows us the whole process of gene expression coming all the way down and all the different ways that eukaryotes can control how much a gene is expressed. And we're going to talk about all of them here. Histone modification, transcription factors, alternative splicing, microRNA, translation inhibition, and degradation of proteins. That's all the different ways that a eukaryote can control which genes it expresses and which genes it does not express, which proteins it makes and which proteins it does not make. Histone acetylation is the first one, modifying or methylating histone proteins in chromosomes to access specific genes in a chromosome. So you remember how I was talking about how chromosomes are kind of like spools of thread, right, that are kind of stacked on top of one another? Here's what I mean. Here are histones, and you see how the DNA is kind of wound around them. Let's just say I'm a cell, and I want to express this particular gene here. What I can do is methylate the protein and change the shape of the protein by adding what's called a methyl group to it. And therefore, I can access now, check it out, now I can access this little piece of DNA, this little gene that I can make my protein with that I want to. Okay, so it's used to access very specific genes in a chromosome. Transcription factors, I mentioned these before. Proteins that bind to specific DNA sequences such as enhancers, which promote transcription, silencers, and prevent transcription, and insulators, which I believe also prevent transcription, but you might have to check me up on that. But anyway, what you have to know is that a cell can use different transcription factors to say, yes, express this gene, or no, do not express this gene. Okay, positive transcription factors say yes, negative transcription factors say no. Sequencing of transcription during development controls gene expression at various stages of development and life cycles, right? So this is so as a person, let's just use a human for example, as a person grows from being a baby to a toddler to a child to a teenager, right? They're going to be expressing different genes at different times as well. And transcription factors are these proteins that are going to be saying, hey, you express this gene now and you stop expressing this gene later. You express this gene now and you stop expressing this other gene, right? So like, for example, you're going to make different hormones, right? You're going to make different hormone receptors as you grow up to be a teenager. And you're going to make different proteins as an adult than you did as a child. And transcription factors are largely responsible for that. Another way is through alternative splicing, okay? What a cell kind of treats as an intron versus what a cell treats as an exon can alter the ways that genes are expressed. Alternative splicing can edit primary RNA transcripts, what we talked about in 6.3, so that it produces different proteins based on which regions are spliced, which are introns and which are exons. So here's a very, very simple picture, right? If this is not an intron, it doesn't get spliced, this is going to produce a different protein than if it is an intron. Then this is going to be the gene, and this is going to be it's going to make a different protein once it goes to the ribosome, right? That's what's called alternative splicing. MicroRNAs, this is fun. A small RNA molecule that binds to a complementary mRNA sequence and it blocks translation, right? So mRNA is one strand, right? Single-stranded. Now, what if you take a little tiny piece of RNA called a microRNA and you bind it to that mRNA, well, then it can't go through the ribosome and that gene cannot be expressed, right? So just say, whoa, pump the brakes, do not express this gene. You can add a microRNA to basically stop translation from occurring, which is kind of cool. And the last two ways, I'm just going to briefly go over them. Cells can also block translation. Okay, they can just stop translation from being taking place by degrading a ribosome, or they can degrade an already expressed gene or a fully formed protein. So after everything is done, the gene has been transcribed, the RNA has been modified, the gene has been translated, and the protein has been folded, a cell can still therefore control gene expression by just saying, hey, protein, just go to the lysosome and degrade and see ya, we don't need you, right? Lots of different ways for eukaryotes to express their, to control their gene expression. Now, 6.6, I'm going to go through this very briefly because I already talked about it. Differential gene expression allows for specialization of cells in eukaryotic multicellular organism. Depending on which genes a cell expresses and which genes a cell does not express, allows it to do different jobs. Okay, so I cannot stress that enough. Here's stem cells, right? Stem cells are really sweet. Very, very, well, I mean, in the last like 20 years, revolutionary applications to medicine, right? So stem cells can become any other different type of cell. They're hence pluripotent embryonic stem cells. They can become any other type of cell. And how do they do that? Well, they receive certain signals to tell them to express certain genes. So that a lung cell produces different proteins than a pancreatic cell. A heart muscle cell is going to produce different proteins than a red blood cell, so on and so forth. Same genome, different expressions for different jobs. Super important. That's what stem cells are. They're unspecialized. They don't have a job yet. Maybe like you watching this video. Cells found in growing embryos and tissues. And they receive signals to start developing into different cell types. They receive signals to start developing into different cell types with specific jobs. I cannot stress that enough. Different cells have different jobs to do, and how they control their different jobs is by the genes that they express and the genes that they don't express. There you go. Okay, here's an immune cell, here's a red blood cell, here's a neuron, here's a stem cell. It can become anything else but that the body tells it to. A cell's phenotype is determined by the combination of genes it expresses and how much they express them. Been over that. I'm just going to move on. We're on topic 6.7, last couple topics. Mutations are any random change to genetic information of a cell or a virus. Mutations, super important for next unit. They are the ultimate source of new genes, largely responsible for the genetic diversity in living things. All new genes come from mutations. New genes are created by mutations, and they're just a random change to a sequence, right? If I got TGT, CTT, GC, ATAC, right? If that's altered in different ways, different letters are altered, that results in a different mRNA transcript, that results in a different protein, and it results in a different trait, and that's really exciting because it can be a really bad thing or it can be a really good thing. All right, but either way, mutations are the ultimate source of genetic diversity, and that is crucial for organism survival and reproduction, and provides a basis for natural selection and evolution. Little spoiler or a little teaser for next unit, Unit 7. It's my favorite. It's the best. Point mutations. There's different kinds of them. All right, and I'm going to ask you to know like 4. Okay, point mutations involve a change in one single type of nucleotide of a gene, and there's 3 different examples of point mutations. So 2 kind of, but anyway. Silent mutations are those that do not affect a phenotype, right? So if my regular gene goes AAG, and then there's a mistake that happens, something changes the DNA and it changes it to AAA, guess what? It doesn't really matter, because if you transcribe it into mRNA and translate it, it still produces lysine, okay? So there's no change here. That's why we call it a silent mutation. It doesn't really change the phenotype. Missense mutations are the point mutations that change one amino acid to another. So if my original codon was AAG and it produced lysine, my codon now, AGG, will produce arginine, or ACG will produce threonine, and that would change as one amino acid, hence kind of why it's called a point mutation in my protein, and that can alter the protein's function. It can, change the protein's shape. We don't really have to know the difference between conservative and non-conservative missense mutations, right? But it's saying that a conservative missense mutation is going to produce an amino acid with the same properties as the original one, but a non-conservative one is going to produce one with different properties, right? So if it changes the amino acid from being non-polar to polar, that would be non-conservative, but you don't have to know that. And then the third type of point mutation is what's called a nonsense mutation. This is interesting. If you change the gene and you change the codon to one that would result in a stop codon, then you're just cutting off the protein, and it just there's no more amino acids after that stop codon. Hence why it's a stop codon. And it results in a very, very short polypeptide. So this can be pretty, this can drastically alter the protein because you're just cutting it off. Speaking of drastically altering a protein, a frame shift mutation. This is a big one. A number of nucleotides inserted. You can add nucleotides or take them away. Those are also different types of mutations into a gene. Is not a multiple of 3. Remember, because it reads a codon is 3 nucleotides at a time. So if you add 1 or you take away 2 or something like that, you can lead to a change in what's called the reading frame of the ribosome, and it can result in a different polypeptide. So here's a good example, right? Adding 2 nucleotides, okay, here is going to alter the protein in such a way. After that insertion, what we call adding 2 nucleotides, every other amino acid is going to be altered after that. It's going to change the groups of 3 that the ribosome recognizes. It's going to change the groups of 3 that the tRNA are going to bind to, and then add the amino acid to. It changes everything after that insertion of 1 or 2 nucleotides, or the deletion of 1 or 2 nucleotides. If it's 3, it's not a big deal, right? It's not going to change the whole rest of the protein. You might be missing an amino acid, or you might have an extra amino acid. It's going to be more like a point mutation. But a frame shift mutation is going to alter everything down the line, okay? Because it adds only 1 or it adds 2, and that's going to change the groups of 3 all the way down the line. Mutations are caused by errors in DNA replication due to mutagens. Mutagens are anything that causes mutations. This could be ultraviolet radiation. This could be anything in a cigarette that you smoke. Toxic chemicals, right? Those are what are called mutagens and cause mutations. And here's the thing about mutations again. Coming up on next unit, Unit 7, mutations can be detrimental, they can be beneficial, or they can be neutral depending on an organism's environment, right? Beneficial means that, hey, it helps it survive and reproduce. Neutral is just like, who cares? It doesn't really help or hurt anybody. Or it can be detrimental, or what's called deleterious, if it causes problems and lowers that organism's chance of survival and reproduction. Beneficial mutations can enhance an organism's ability to survive and reproduce, and therefore will get passed on to subsequent generations. So the fun fact, a polar bear's transparent fur is a result of a genetic mutation that was passed down from one generation to the next, and eventually resulted in a new species. Really cool stuff. We'll talk about that next unit. Speaking of beneficial mutations, bacteria, antibiotic resistance. How do prokaryotes become genetically diverse? This is basically how. Bacteria can receive mutations that make them antibiotic resistant, greatly increasing their ability to survive and reproduce. So in certain bacteria, since they reproduce every 20 minutes and they are kind of bad at copying their DNA, they make a whole bunch of mutations, and some of those mutations are exactly what they need in order to help them survive and reproduce. And that's the root of what we call antibiotic resistance, right? So you get strep throat or something like that. You take antibiotics for it, right? But what if that bacteria doesn't respond to the antibiotics that you take? That's a scary thing, and this is where that comes from. This is a real thing that's happening in the medical field today, is that we are dealing with antibiotic resistant bacteria. MRSA, methicillin resistant Staphylococcus aureus, if you've ever heard of that. That's a big deal. These advantageous mutated genes can be passed on to their offspring or horizontally to other bacteria. So bacteria, as I was saying, have high rates of reproduction every 20 minutes in some strains of E. coli, and have high rates of mutation. Therefore, they can evolve very quickly. We're going to understand this better next unit. Application of antibiotics makes antibiotic resistance more likely to survive and reproduce, right? So if some bacteria have a gene that allows them to say, hey, I'm going to make this protein and your antibiotic does nothing for me anymore, that is obviously a really big advantage for that bacteria. It's going to survive and reproduce better than other bacteria without that mutation, right? And that's where this comes from. So now, bacteria, they can pass that down to their offspring, but they can also pass the gene on to their friends, I guess, to other bacteria, so that everybody can become antibiotic resistant. It's really diabolical. And there's 3 ways that can happen. Transformation is 1, when prokaryotes alter their genotype by taking up and expressing foreign DNA from their surroundings. What? Yeah, they can just kind of absorb a plasmid. Remember plasmids? I said remember that. Plasmids are circular pieces of DNA. It can just kind of absorb that from its environment. All right, and there's a very special lab that we can do in AP Bio. I hope you do with your class, where you make bacteria absorb a plasmid and alter its genotype and phenotype. It's really cool. So transformation is 1. Transduction is when phages or viruses infect bacteria and carry prokaryotic genes from one cell to another. So here's transduction over here. So here's a potentially beneficial gene. It's carried over from one bacterial cell to another via a virus. That's called transduction. And the third type is called conjugation, when bacteria temporarily join and directly transfer DNA through a tube. Here's a very, very silly comic, right? But it's kind of how it works a little bit. Like, hey, you want to become a superbug antibiotic resistant? Take this gene. Like literally, it creates a tube between one bacterial cell and another. It says, here, take this gene. And now you're antibiotic resistant too. Absolutely bonkers. I don't know. That's crazy to me. But how this is possible is through, as I said before, plasmids, small rings of independently replicating DNA molecules. It's not part of the bacterial chromosomes, and they can be transferred through conjugation and through transformation, which is absolutely crazy, right? Just, hey, little piece of DNA, send it on to a friend, and now you're back to antibiotic resistant too. Just mind-blowing. All right, last topic. Here we go, 6.8. This is a big one. Genetic engineering is kind of uses the structure of DNA, structure of RNA, and kind of alters. This is how we control this whole process of gene expression. It's the direct manipulation of genes for practical purposes, taking advantage of DNA's properties to achieve benefits for people, right? And this picture is kind of misleading, right? You can't just inject a tomato. Now it's red. That's not how it works. It can be used to optimize crop yields in criminal law and medical and biological research. Yeah, this picture I just found on the internet. I'm like, I typed in genetic engineering and it came up, so I just put it up. But it's misleading. All right, just take a note of that. But yeah, genetic engineering. We're going to talk about 4 different types of genetic engineering. One is called DNA cloning or transformation. Hey, that's what we were just talking about, making bacteria kind of absorb a plasmid from their environment and making it express that gene. And that can be a good thing, right? It's not always about like, hey, now you're resistant to antibiotics. It could be like, hey, make more of this insulin. Here's the insulin gene. Make some insulin so I can extract it and, sell it and, help people with their diabetes, right? That kind of thing. So transformation is cloning DNA using a vector organism to express a desired gene. So we're going to make bacteria express a gene. Foreign DNA can be inserted into a bacterial plasmid, which is then expressed and replicated. Hey, and how do you get a specific gene? Hey, like I want this specific gene. I want this insulin gene to be expressed. Scientists have developed what are called restriction enzymes that cut genes at very, very specific points. And I know in my class, maybe you do it too. We study a few different restriction enzymes, and we kind of, pretend we're little restriction enzymes and cut up a piece of DNA based on where the restriction enzyme tells it to cut. But anyway, this is a lab that hopefully you do in AP Biology. You've done with your teacher. This is the PGLO Bacterial Transformation Lab, where you treat bacteria with a plasmid that allows it to glow under ultraviolet light, and it makes it antibiotic resistant as well. So if you don't know what this means, hopefully you will sometime soon. And if you got questions about how this lab works, I'd be happy to explain it to you. Just let me know. But I'm going to try to wrap up this video because I know I've been talking for a while. Gel electrophoresis is a second type of genetic engineering, a type of biotech where we can separate DNA fragments based on size using a polymer gel and an electric field. So gel electrophoresis, running a gel, as they call it in the business, is really, really cool. You can cut up DNA into very specific pieces using those same restriction enzymes as I mentioned before, and then you can set it up in such a way that, DNA is negatively charged. It's an acid. It's kind of cool. So therefore, it is, it can be moved around using charges, and that's what gel electrophoresis does. There's a negative end of a chamber and a positive end of a chamber. And DNA is, once that current kind of runs through it a little bit, electrical current, it's going to move towards the positive end. And the bigger pieces are going to not move as far towards the positive end as the smaller pieces, and therefore you can get a picture of somebody's DNA cut up with the same restriction enzymes in a gel, and in just like this nice picture here, and I'll show you what that looks like in a second. But DNA molecules yield the same size fragments with the same restriction enzymes, and it enables DNA fingerprinting. And this is what a gel looks like. Whoops. This is what a gel looks like, right? So here's 2 different pieces of DNA that were run through a gel, right? So here's a negatively charged end and a positively charged end. And if somebody's DNA is cut with the same restriction enzymes, it can result in the same pattern of these DNA fragments moving from the negative end to the positive end. And this is how DNA fingerprinting works, right? You can compare the gel of, I don't know, the DNA found at a crime scene versus the DNA of a suspect, right? And if they're not the same, then it's not the person that did the crime, right? So this has exonerated many people that were wrongly accused of crime. It's kind of cool. It's DNA fingerprinting, right? And it's just using the properties of DNA, right? That you can cut it into very specific pieces and send them out towards each other and spread them out so that these pieces result in different spots or end up in different spots in this gel, and you can get this nice picture of how DNA is compared between two individuals. It's really cool. All right, second to last thing I'm going to talk about, polymerase chain reaction. You might have heard of this when we were talking about like COVID tests, for example. PCR COVID tests are used to see whether or not somebody is COVID positive. PCR copies a DNA fragment or gene into thousands of times, manipulating DNA replication to produce billions of copies of a gene, right? So if I want a very specific gene, I can cut it up with restriction enzymes, and then I can put it through this machine called a thermocycler and add specific primers and DNA polymerases, a very special DNA polymerase, in order to make billions of copies of gene in a very short amount of time. So basically how it works is it takes a gene, it denatures it, spreads it apart, uses primers, just like we talked about with DNA replication before, primers to anneal to the template strand, and then DNA polymerase, a very special DNA polymerase called Taq polymerase that is heat resistant to make new DNA strands, and it has to be heat resistant, okay, because heat, increases reaction rates. And DNA polymerase under high heat is not going to be able to copy DNA, so it uses a special heat-resistant DNA polymerase to create new strands. So basically, PCR making billions of copies of a gene. That's what you need to know. And then the last slide here, so page 72, is about DNA sequencing. The last thing that we can talk about is that machines can determine a gene's complete nucleotide sequence by adding a complementary strand, one nucleotide at a time. Right, so you can figure out an entire genome. Like, oh, is it an A? No. Is it a T? No. Oh, it is. Oh, it's a C. That means that the G is the next gene. Kind of it. That's a really simple explanation of it. But DNA sequencing opened up new possibilities in understanding evolution. We can understand our human genome and compare it to that of a chimpanzee. We can compare it to that of a banana, and see how many. Yeah, banana. And see how many nucleotides are the same, and we can we're going to get into this in evolution. We can study phylogeny and a species' evolutionary history based on its gene sequence. Whew! All right, that was Unit 6. I hope that wasn't too long. Let me know if you have any questions. We will see you next time for Unit 7.